Okay. So, in bacteria, DNA is organized in a circle. The circle is called a plasmid. So, in our DNA and any DNA, there's genes. And genes are just a section of DNA that codes or is the recipe for a protein. So, in this case, we have our DNA here, and we want this section that codes for insulin. So that's the recipe for insulin, how to make insulin. So the first step is you want to take a restriction enzyme and cut this piece of insulin out, because you're trying to get that strand of insulin, because that's what you want to work with. That's the recipe you want. Then you take the plasmid out of the bacteria cell. You cut it with the same restriction enzyme. Why is that important? Why do you need to cut it with the same restriction enzyme?
Just cut it in the same place?
It's not the same place.
Same length?
It's not the same length either. So the ends are like match. So it's like a puzzle piece.
So it cuts it the same way.
It cuts it the same way. Not the same place, but the same way.
Gotcha.
Because if you don't cut it the same way, this piece won't fit in, which is what we want to do. We want to take this piece out now and put that in. Does that make sense?
Mm-hmm.
So we cut with the restriction enzyme, we put the insulin into the plasmid, then we put the plasmid back into the bacteria cell. The bacteria, which in this case it's E. coli, but the bacteria then will do normal cell function, so it will do mitosis, so it will divide, and all the new bacteria cells produced by it will have that insulin gene in it, because it copies its DNA and divides and does all that. Well, now E. coli, when it's going through transcription and translation, it's going to make insulin, because in its plasmid, in its genetic information, the instructions for insulin are there, and so it's going to go through and follow through like normal and make insulin.
Can we review transcription and translation real fast one more time? I know it's from DNA to RNA, and what happens there again real fast?
It's not a really fast answer. I could go over it after this. Okay. So the bacteria starts making insulin, and then that's the insulin that we use for drugs, like for diabetics when they go to the pharmacy and pick up insulin shots. It's because we have laboratories making tons of insulin. Does that make sense?
And
Okay.
that's genetic.
Engineering.